cougar light, clearing the malignancy. Target acquired. Malignancy cleared.
In this video, I'm going to be talking about the mechanism of tunneling nanotubes between immune cells. So from immune cell to immune cell. So TNTs are thin, actin-rich membrane bridges between cells. So they allow direct exchange over distances, such as various different cargoes, such as mitochondria. They even transfer DNAs, RNAs to each other. So immune cells that commonly use TNTs are dendritic cells, NK cells, T cells, B cells, and macrophages. TNTs can be short-lived, minutes, or persist in hours. So this goes to show that what you eat has a huge effect on your immune cell, and depending on how much you eat or what you eat, how long the TNTs are going to last, which is minutes or hours. You go like a long way without eating anything that could support TNTs for immune cells, then that would leave you at a huge disadvantage.
This is how cancer evades the immune system. Cancer uses PD-L1 to disable T cells. But now we're fighting back with nanotubes. These nanotubes deliver a powerful signal, SCAR5. And here's the twist. Green tea. SCAR5 activates T cells, while green tea boosts their function. Together, they attack
calcium ions for rapid intracellular signaling. Also, second messengers such as CAMP IP3, they can propagate activation signals. They even transfer reactive oxygen species to modulate redox state of recipient cells.
Mitochondria, the powerhouses transferred between cancer cells. Tunneling nanotubes enable direct transfer, fueling tumor survival and spread. This connection is their weapon.
Immune cells, such as T cells, they need IL-2 to thrive. Because our cancer cells, they recruit Tregs or Tregs that steal IL-2 away from immune cells. So helping build TNTs between immune cells can help them transfer IL-2 to each other, especially in a situation where immune cells are struggling to battle cancer cells. And if they are low on IL-2, then the immune cell can transfer IL-2 to another immune cell. Therefore, that's like a teamwork. It's
in the
like
depths of cancer cells, a
immune
secret
cells
network is forming. Tunneling nanotubes connect these tumor monsters, transferring
tumor
mitochondria from one
microenvironment
tumor,
that can be very hostile against the immune cells. Pathogens, they might
transfer
exploit
stops.
Endosomal cargo transfer that spreads between immune cells, so which is actually beyond the scope of this video. Also, what else is transferred from immune cell to immune cell is exosomes and microvesicles. Also,
Cell, under stress, trigger abnormal responses. Tunneling nanotubes formed under mitochondrial stress are now out of control.
Which would make sense.
Magnesium supplementation may help regulate mitochondrial function, restoring balance, one ion at a time.
Energy to do
Diesel and
things
means
like
to trigger apoptosis in empty or
Perforin,
cancerous cells,
Granzyme
but
B,
perforins, proteins that punch whole cells, are too weak. Without them, Granzyme B
need a lot of
can't enter.
energy
Cancer
for
cells
that.
survive.
For
Enter
example,
Goku, boosting Granzyme B and
the
perforin
Perforin is
production.
like a
With
tube
enhanced
that
perforins and
attaches
Granzyme
to a
B,
cancer
the immune
cell,
system strikes.
and then the Granzyme B goes through the tube and then into the cancer cell, making the cancer cell go through apoptosis or programmed cell death. And immune cells need a lot of energy to be able to do this kind of attack mechanism on a cancer cell. So that's why it's important for mitochondrial DNA to be transferred from immune cell to immune cell. Also, lysosomes can be transferred.
Key to iron regulation.
Chroma
DNA holds genes for lysosome function. Immune system genes are failing. Time to fix this!
Can be very stressed.
Sulforaphane opens chromatin. Lysosome genes activate, iron handling restored. Genes and iron now in harmony. That's how you heal a cell.
Cargo. So one mechanism they do that is through endocytosis. The recipient immune cell engulfs the vesicle or exosome, or supposedly cargo that's being transferred from immune cell to immune cell. The way endocytosis works, so that the immune cell can receive the cargo through the TNT, is so the CD14 is attached to the Toll-Like Receptor 4. So the TLR4
When CD14 is cleaved, TLR4 can't engage with TRIF, meaning immune cells fail to activate. Without this signal, cancer cells go unopposed. But bone broth supports intact CD14, allowing TLR4 to engage TRIF and reactivate immunity. Proline, a key amino as bone broth, helps maintain CD14.
Is possible. Also, things can cleave off the CD14 though. If that occurs, then the Toll-Like Receptor 4 cannot interact with TRIF, and therefore lack of that interaction means no endocytosis. So theoretically, that means that the immune cell cannot receive a cargo through a TNT from another immune cell. Okay, other cargo that can be received by immune cells are contents like mRNA or microRNA, altering transcription. So sounds like if immune cells through TNTs can help each other even at a DNA level, because mRNA alters transcription, and transcription occurs at chromatin. Inside the chromatin or the DNA.
Open chromatin is driving cancer.
Also help each other by restoring ATP
Silence
levels
oncogenes.
in stressed immune cells. The integration into existing mitochondria, the
Silence
donor mitochondria
oncogenes.
being sent from another immune cell, can fuse with the mitochondria of the receiving immune cell. Another really good thing about TNT transfer is that it helps amplify the activation of T cells. Also helps antigen presentation. Basically, antigen presentation is where the T cells have to be trained by the antigen-presenting cells, because the T cells are naive and they don't need what to do, and they need guidance.
CD80 and 86 restores T cell response. With
Also,
proper co-stimulation and targeted
there's a risk
destruction,
of
immunity.
pathogen spread because some viruses, like HIV, exploit TNTs to move between immune cells. Immune cells, especially T cells and NK cells, they expend enormous amounts of energy in the TME. So the energy loss happens through several mechanisms. First is high metabolic demand. Activated immune cells switch to glycolysis, even when oxygen is present, to rapidly produce ATP, and also continuous cytotoxic activity, releasing perforin and granzymes. So basically, perforin and granzymes, immune cells use that to attack cancer cells. So basically arming themselves with weapons, which would be the perforin and granzymes, and the actual activation of immune cells uses up a lot of energy. And so activation of immune cells is through glycolysis. Okay, so it's very important that immune cells through TNTs, that they get their mitochondria for energy, because if they don't, then they can't arm themselves with perforin and granzymes, and they can't activate. So some good nuts here to help with that is pectins and almonds. Pectins and almonds, they help mitochondrial proliferation or mitochondrial numbers for immune cells. So since immune cells are high energy demanding cells, pectins and almonds, they also protect the mitochondria as they are being transferred from immune cell to immune cell via the TNTs. Okay, here's another reason why it is critical and crucial that T cells are getting their mitochondria, especially in the TME or ECM, is that tumor cells, they consume glucose and amino acids in the microenvironment. In fact, they actually outcompete immune cells, because immune cells also need glucose. But if tumor cells are taking up all the glucose, then that's going to weaken the T cells. So at least T cells have some kind of help and support with mitochondria, also with nuts like pectins and almonds. Tumors also take up glutamine, arginine, and tryptophan, depleting T cells from much needed energy. So that's it for the video. Like, comment, share, subscribe, and I'll see you in the next video.